Gene cloning tools, expression vectors for bacteria. This session will give you a very brief overview on expression vectors, and the focus will mainly be on those that are used for bacteria. But one can use expression vectors for higher eukaryotes as well, for example, mammalian cell lines, insect cell lines, etc. So let us look at the learning outcomes of this session. When a cloned gene has to be used for production, that means you just don't want to amplify the cloned gene, but you want that the cloned gene be expressed into its corresponding product. Then one has to use expression vectors, and those expression vectors have to be suited to the host organism. The expression vector, apart from having the conventional specifications of a cloning vector, should also have a promoter, ribosome binding sequence, and terminator. The promoters have to be chosen with care, especially the promoters have to be such that they are active in the host. That means they are recognized by the machinery of the host organism, as it would decide whether expression will happen and at what rate the expression will happen. The construct of the expression vector can be modified based on the need. So primarily, what is expression vectors? Say for example, you are isolating a DNA from an animal cell, and from that DNA you isolate a gene of interest that gives rise to a protein that has a certain demand. Now that gene which gives rise to a product that is useful is cloned in a vector, and that vector is then transfected into a bacterial cell. Now that bacterial cell should be able to transcribe the gene of interest into its mRNA, and that mRNA in turn has to be translated into a product, that is a protein. Now, you must understand that you are trying to express an animal protein in a bacteria, which is why the vector should have a promoter, or the vector should be such that the bacterial system is able to recognize the promoter and express the gene, although the gene is from an animal cell. So that is the essence of the construct of expression vector. It has to be suited to the organism in which the product is going to be synthesized. Now, in order for a product to be formed, what is required is that upstream of the gene that has to be transcribed and translated, upstream of the gene should be present a promoter region. Now, promoter region is a region that is recognized by RNA polymerase, and RNA polymerase on binding to the promoter region can initiate transcription. Also importantly, once you have the transcript being formed, the transcript should have a region where the ribosome can come and assemble. So you have what is called as a ribosome binding site that when in a transcript would be recognized by the ribosome, and the ribosome will assemble at this point and start translating the transcript. So essentially what is required is that this gene of interest should be transcribed into a transcript, and that transcript then should be translated by the ribosome machinery. Now, also what is important is, suppose you have a promoter, then one should have the terminator for the transcription as well, because otherwise you would keep having the RNA pol synthesizing more and more of RNA which is not related to the gene of interest. So therefore, one has to terminate the transcription so that you get a transcript of the gene of interest only. So therefore, an expression vector should have a promoter, should have a ribosome binding site, and should also have a terminator sequence. In E. coli, so when you want to use an expression vector in E. coli, the promoter that the E. coli RNA polymerase recognizes is a -35 box. So you have a sequence at the -35 region upstream of the gene of interest, and you have a -10 box. So this is the Tata box, and this is the TTGACA box. If you have both TTGACA and TATAAT with interspersed within it 17 to 19 base pair, then this is considered to be a promoter which would definitely be binding to RNA polymerase with great affinity to transcribe the gene of interest. So this is considered to be a strong promoter. Now, therefore, the understanding is that if you have the expression vector, you have the promoter, you have the ribosome binding sequence, and here is where there should be a restriction site, and cutting the vector with the restriction site, a gene of interest has to be inserted at this point. So then the gene of interest is flanked by the promoter and ribosome binding sequence at one side and flanked by the terminator at the other side. So then, if this is the case, the promoter has to be a promoter that is recognized by the RNA polymerase of the host. So it can transcribe, form the mRNA, and that mRNA should have a ribosome binding sequence or site that can be recognized by the host ribosome machinery to form the product. Now, when you have such a system, as you can see, you cut the vector, insert the foreign gene. Okay, then this is what is called as a cassette vector. Okay, so when you have between the PR and the T a gene of interest inserted, then that is what is called as a cassette vector. So if one uses a strong promoter, but obvious, the number of transcripts that will be formed would be high, and hence you will have a high amount of protein being or products being formed. While if you have a weak promoter, then you will have a relatively lower, fewer number of transcripts formed and a lower amount of or number of proteins being formed. So depending on what is the requirement, you could use a strong promoter or you could use a weak promoter. Now, what is also interesting and a point to be noted is that the genes can be switched on and switched off depending on the need. So that would be essentially dependent on the promoter and whether the promoter can be induced. That means if there is inducible gene, then it means that the gene is normally switched off, but in presence of a regulatory chemical, RNA pol will effectively come and bind to the promoter and transcribe the gene. So then what we understand is that this regulatory chemical is switching on the transcription of this gene. So therefore it is called as the inducible gene. On the other hand, you may also have a repressible gene. So a repressible gene is such that it is normally switched on, that is, it is continuously getting transcribed. But in presence of a regulatory chemical, what can happen is that the regulatory chemical can switch off the transcription. So this is a repressible gene therefore. So you can either have it getting induced, that is switched on, or you can have a switched on mode of gene switched off. So basically, the understanding is that in expression vector, when you put the gene of interest transcription can be regulated. And that regulation is dependent on what kind of promoter one is using. Now, let us take, for example, the plasmid GEM3Z. Now, GEM3Z is basically a modified PUC8. So, as you can see, it has a size of about 2750 base pair. This is a very good characteristic of the plasmid because it means that one can have a good sizable insert possible within it. But you can see that there is an ampicillin resistant gene. Here you would not have any restriction sites, but you have a second selectable marker, which is LAGZ prime, and the restriction sites are present within the LAGZ prime. But since it is an expression vector, you can see that flanking the LAGZ prime at one end is a T7 promoter and the other end is a SP6 promoter. Now here there is a catch. Ideally, you should have a promoter that is that of the bacteria. Suppose you add this or transfect this into E. coli, then the E. coli RNA polymerase should recognize this. But here you can see that this is a T7 phage promoter, which means that a T7 RNA polymerase, that is an RNA polymerase of the T7 phage should be able to recognize it. Or it may be the case that a T7 promoter being very strong, therefore its use is advisable. In such a scenario, the E. coli that is used should already have the T7 phage within it so that its RNA polymerase can recognize and carry out transcription. So T7 and sp6 promoters are fast promoters, but they are strong promoters, and hence you can see how the expression vector has been designed to ensure that you have a high rate of transcription taking place. This can be used in E. coli, but with RNA polymerases of the respective phages. So let us make the conclusions. Expression vectors that are to be used in E. coli or a bacteria should have a promoter, a ribosome binding site, and a terminator. Now you must understand that when you consider bacterial systems, RBS basically stands for, or RBS basically is a Shine-Delgarno sequence or its equivalent. The RNA polymerase of the bacteria should be able to bind to the promoter region to initiate transcription. The choice of promoter is definitely very important. Depending on whether the promoter is strong or weak, the rate of expression and the transcript levels vary. The promoter can also be regulated. The ribosome binding site should also be suitable for the host organism's translation machinery. Therefore, expression vectors are constructs that enable the gene of interest to be transcribed and translated so as to get an engineered bacteria capable of not just amplification of the gene of interest, but production from that gene of interest. Thank you.